So, I'm just going to find the page. Hold on 1 second. Okay, so I want to talk a couple of things about your homework, and then we'll get started. So the biggest thing I want to remind you about, I think all of this should be fairly straightforward. There's nothing exciting, but we're going to start hitting your project really heavy. Do you know what I mean? Meaning that there's a lot of stuff that are going to be turned in. And so you need to sort of make sure that you're on top of that. So a couple of things. Your annotated bibliography is due a week from today. Okay, a week from today. So actually, if I can have you pull out that research, thing that I had you print out at the very beginning of the semester. So the directions for the research project, pull that out. And if you don't have it handy, be by someone that does. So be on page 3. Okay, so a couple of things. What you're turning in is task 4, and the directions for all of that are on this page, right? Page 3. So a couple of important things. I think you already know this, but you're going to have 4 sources. One of them is a journal article. Only one can be a website. So some of you, we talked about, I had too many websites, and I need to make sure that I get rid of one and have some other source. So 1 journal article, 1 website, and then 2 other sources. Okay, and if you don't want to have a website, that's okay. But you can't have any more than 1 website. All right. And then look maybe halfway down, like let's say the page, it says after each citation. So what you'll have is the citation at the top of the page. The citation should be in APA format, and it should follow like, you should be citing properly using your APA booklet that you got from the library. So, if it's a journal article, you follow that example and make sure that you're citing it properly. And then after that will be a half-page summary of the article. Okay, it has to be of just that particular article, not of your entire project. Just a summary of that particular source. And so make sure you don't state it. Some of you are saying things like, this article by John Hopkins tells me, I already know that it is a summary of that article. You don't have to tell me what it is. Just launch into the information. And it says sort of halfway down, make sure that you have chemistry-related topics. So some of you didn't have very much chemistry in the summaries that I read. They need to be in there. Not every source has to focus on chemistry, but by the time I've read all 4 of your summaries, I should see that you have chemistry in your topic. Okay, that's really important. Make sure that your sources are not what your partner's sources are, that the information that you're researching it's individual. It's for your part of the project. So some of you are going to look at neurotransmitters, some of you are looking at enzymes. Just make sure that you have that information somewhere in there and that it's specific, not like neurotransmitters are affected by this drug. Well, which drug? And how come, what neurotransmitter are affected and how are they affected? Do you get more of them? Do you get less of them? Make sure that the details are in there. Okay, so turn with me, all the directions are there. Oh, one more thing, you're also going to turn in copies of the sources and they should be highlighted. Okay, so what I'd really like is your citation and your half-page summary. You can staple it to your source, that way I know which ones go with which, and make sure that the source is highlighted. I'm also going to check that you're not plagiarizing, so be sure that it's in your own words. There's no quotes, nothing like that. So look at the score sheet. The score sheet for this is on page 7. So it's 23 points. It's kind of like a really big quiz. And so it's like 5 points for each of the sources and then plus a couple other things. So each partner must have 4 sources minimum. I'm literally going to staple this score sheet to your annotated bibliographies. This is how I'm scoring it. So my advice to you is that you look through this carefully and make sure that you're not missing anything as you put your annotated bibliographies together. So we already talked about the types of sources. You guys can see that there. But for the summaries, they need to be typed. If they're not typed, I'm not going to accept them. So be sure that they're typed, double-spaced, 12-point font, half a page. So that's like 100 to 150 words. Please don't make me count, because I will count if they're too short, and I will take points off if they're not short or if they're not long enough. Okay, so be sure that you have at least 100 to 150 words. Written in your own words, don't copy. No direct quotes. Don't list the information. Make sure that chemistry is included. You talk about how the disease works or how the drug works, or the treatment, whatever it is. I need to see some chemistry in those summaries. The information should be organized and easy to understand. So this shouldn't be like, it should be well written. And if you feel like you have a hard time writing, then take it to the Writing Center. They'll look at it for free. They'll check to make sure that your citations are in APA format. So if you're not sure, take it up to the Writing Center. They're on the 4th floor. You were there when you picked up the APA booklet at the beginning of the semester. So it should be written in third person. There should be no I's, we's, and you's. So you wouldn't say something like, you know, you shouldn't take this drug if you're pregnant, but you might say a person should not take this drug if they're pregnant. Also, no contractions. So be sure there's no contractions in there. Correct spelling, grammar, punctuation. So like, I shouldn't get halfway through your summary and feel confused and have no idea of what you're talking about. And that just means it's not organized. Okay, so I would have a second person read your summary, and if they can understand it, then it's probably fine. You know, I think as the author, like, you know what you want it to say, and very often it feels like it makes perfect sense to you, but that's because you're the one writing it. Do you know what I mean? And it may not make sense to someone else. So make sure that you might have another person actually read your summaries. Okay, and then this highlighting part we talked about, and then the APA part. So I would really use this as you put these together so that you make sure you're not missing anything. This is a good opportunity to get, you know, a perfect score, or at least close to it. So these are due in a week. That means that on Wednesday, we're setting a lab up. So we're gonna make soap on Wednesday, and that will take a short period of time. So we'll have a couple things to do on Wednesday, but for sure we'll probably be done, I'd say at least an hour and a half early. And then that gives you all of that time to be working on your annotated bibliographies. Make sure you're pulling those together. You have a lot of the pieces. Like, you've probably written at least 2 summaries for me so far, and maybe you have to fine tune those, and then maybe you have 2 more to do. You know, it shouldn't be too horrible. Do you know what I mean? But there is some lab time to work on that on Wednesday. And then what's coming after that, and it seems kind of far away, like 2.5 weeks, but that's going to go by faster than you think. And that is the rough draft of your summary. So I want to talk about that for a second. You and your partner are going to put together a 4-page summary, and it will include a title page. It'll have an introduction. It'll have a conclusion. It will have citations in the body of the paper, and then at the very end will be a references page. And so that is due in 2.5 weeks. So I know that seems far away, but you may want to be thinking about that, because this requires you and your partner kind of getting together. So if you feel like your annotated bibliographies are solid and done, then you should take Wednesday to start working with your partner and getting that summary together. Do you know what I mean? So we'll have some time this Wednesday, and we'll have some time next Wednesday. So you're gonna have 2 lab days that give you some time to work in the library, to get together with your partner, to start working on this part of the project. So that summary needs to visit the Writing Center, and you and your partner need to go together to go to the Writing Center. This is worth points, so if you can't make it, I forget how many points it is. It's less than 10. It's like 4. Actually, I don't really remember. But anyway, let me tell you. It's 4 points. So if you go, great. If you don't go, then you're just losing those 4 points. But it's for them to help you make sure that everything is cited properly, that your summary makes sense, that it has good information, that it flows. So you need to sort of get that summary to the Writing Center, and the later you wait, the longer you wait, the more crowded they're going to be. Because people are going to start having like final papers that they're turning in, and they're going to start to get really crowded up there. Chem 2A has got their project, they're going to get in there. So my advice is to go to the Writing Center sooner rather than later. Okay, that's my advice. And so those are the big things that are coming up. The directions for the summary, just so you know, let me tell you where they're at. So they're on, let's see here, page 4. So flip back to your page 4. So the summary is task 6. You and your partner are going to make a summary. It'll have a title page, 4-page summary, introduction. It'll have good chemistry in it, in-text citations, and a conclusion. In-text citations mean you're not quoting, but you're telling me where your information is coming from. Do you know what I'm saying? So if I said, you know, this drug increases serotonin by inhibiting its reuptake, that should be coming from somewhere. And so maybe after you put the author and then the year. And there are directions in your APA booklet for using in-text citations. Okay, so make sure that you're doing it properly. And we'll talk a little bit more about those as we get a little closer. But mostly, I just want to remind you that that's coming. The score sheet for the summary, if you're looking for that, is on page 8. And so it's 26 points, this summary. And so again, it's like a very large quiz. And you know, you want to just make sure that you do a good job for it. So just want to make sure that you all are sort of aware of what's coming up, okay? Any questions on that? So annotated bibliography in 1 week, and then the summary, the rough draft of the summary is due after that. It is a rough draft, so it doesn't have to be perfect, but the better you can make your rough draft, the more feedback that you'll get from everyone else in the class, and so it'll make the final product better. Any questions? Okay, so you have lab time, right? Lab time this Wednesday and next Wednesday. So however you want to sort of organize that, maybe you spend one day with your annotated bibliography, and then maybe next Wednesday you're working on the summary. Whatever you like, but it's just coming. Okay, it's going to be coming sooner than you might think. Okay, so then let me think. Anything else I need to tell you about? No. Okay, I'm going to put you together with your partner. We're not going to start the lab just yet. I'm going to hand back one of your homeworks, and then I want you and your partner to sort of compare your homework sheets, see what you came up with, then we're going to go over it, and then we'll get started on the lab. So this lab is due on Wednesday, and I just want to say a couple of things. You and your partner might check out the post-lab, do you know what I mean? And think to yourself about answering those questions before you go. The other thing is this lab actually involves calculations, and I'm going to check your calculations before you go today, okay? So keep that in mind. We'll talk more about that in a second. So I'll put you together with your partner. I'm going to give you a homework, and then you guys can compare those. So Gabby and Stephanie. You guys are like right down the street. Okay, Rosie and Zach. Oh. And then Hannah and Larissa. This is over there. Then Katria and Kat. Those are next to each other. Then Courtney and James. These are right there. Jackie and Alyssa. Go. Then Amy with a P and Leah. You guys are next to each other. Anastasia and Jay. Jay's over here. Amy with an L and Amy with an F. So, right here. Then Yelson and Chris. You guys are across from each other. And then one group of 3. So Dylan, Sarah, and Cheyenne. Okay, so go find your partner. I'm going to bring you your homework, and then you guys can start sort of comparing what you have.
Oh, hey Carrie.
Idea?
That's fine.
That's fine. That's fine. I'm going to get those back to you. Yeah. I'm sorry. Let me just add it.
We'll see where they live here. Outfit was like triple size. What do you want to share? Oh, there you are. Oh, okay. I'm just we're pretty good together. Oh, you were? Oh, okay. It's okay.
I'll write it down.
Oh, okay. I but it's more soluble. Because I put syrup acid here.
Oh.
It looked like she was saying
Okay, good. I'll write it down. That's
I'm
nice.
gonna pause this for a second.
Yeah. We start new stuff today. So let's start with page 99. Pull out those pages and let's go through those really quick. So Gabby, can we start with you? Which fatty acid, the palmitic or stearic, will be most soluble in hexane? Palmitic acid. Let me ask you about hexane. Is hexane polar or non-polar? Hexane is non-polar. So whoever should be the most soluble in hexane should be the most non-polar. So who should that be? Yeah. So your question would have been maybe make more sense if it was, say, water, but hexane is non-polar. So who should have the highest melting point? Stephanie. Stearic acid. Good. How come? Yeah, it's bigger. Yeah, it'll have more intermolecular forces. Okay, so for number 2, the first one says, which has the lowest melting point? So this one's for you, Courtney. A, B, or C? C. Good. Okay, and then James, which one has the highest melting point?
A.
Good. And then which is fatty acid is of mostly animal origin? That's for you, Yelson.
A.
Yep. And then D, which fatty acid is mostly of vegetable origin? Chris.
C and D.
Yeah, B and C. And then are any of them omega-3 or omega-6 fatty acids? Alyssa? C. C, and which kind is it? Omega-3 or omega-6? Good. And then what type of double bonds are in B and C? Jackie? Cis, good. And then when you write your hydrogenation reaction, so a couple of things. If you're writing it for C, like, you have your fatty acid, but what should be the other reactant here? Like, what else are you adding to it when you do hydrogenation? Hydrogen. How many hydrogens? 2. Good. That's really important. And then what do you need over the for a catalyst? Okay, so maybe like platinum. What else? Pressure and heat. I just said that. Okay, there it is. I gave it to you. And then what should you see on this side? What will be the difference on the product side? It should be totally saturated, right? So no double bonds. You can draw it exactly the same, just not double bonds. And so this is for H. Patria, did we just do or not? I can't remember. No, we didn't. No. Okay. So before hydrogenation, compound C is a liquid, and after hydrogenation, it is a solid. Good. So like it's going from unsaturated to saturated. Okay, so Kat, which of the above molecules has the highest melting point? A, B, or C? A. A, good. And which one has the lowest melting point? Hannah? C, good. And which one is most likely a solid at room temperature? Larissa? A, good. And in structure C, label the bonds that are cis and those that are trans. So which ones are the trans ones? The top or the middle ones? The middle ones, right? Okay, good. And if I ask you to draw something like, this, 18, colon 2, delta 9, say something like that, when you draw them, make sure they're in the cis configuration. Okay, that's really important. And make sure too, when you start to draw them, you start with your carboxylic acid, right? That is carbon 1. And then you go in that direction. I think some of you are counting in the opposite direction. Okay, which triglyceride can undergo hydrogenation reactions? So, Cheyenne. This is E.
B and C.
B and C, good. And then when it asks you for a saponification reaction, what does saponification mean? Yeah, so you're taking that triglyceride B and you're adding a strong base. What should you get? So maybe I'm adding like potassium hydroxide or sodium hydroxide. And what do you get on the product side?
A glycerol.
Yep. So you should have your glycerol. And then what else?
3 carboxylic salts.
Yeah, and so maybe they look like this. And then whatever this part is. And I actually want you to make sure that the carbons are right. Does that make sense? And the double bonds should still be there if there's double bonds. So like in B, you'll have like 2 that are saturated and then 1 that's going to end up being unsaturated. But they should look like this on the end. Does that make sense? So make sure that it's a carboxylic acid salt. Okay, good. And then that brings us to page 10. Oh, I'm so dirty. How did that happen? Okay, it's fine. Okay, so page 102. This is sort of what I mean by being able to look at lipids and decide what type of lipid is it. And some of you are giving me, like, for types of lipid, membrane lipid. And that isn't what I'm thinking of. So what are the classes of lipids? Like, if we were going to talk about a class of lipids, what would they be? Give me some examples. Like fatty acid counts. What else?
Wax.
Wax. Good. What else?
Triglycerol.
Triglyceride. Good. What else?
Phospholipids.
Good. But let's be specific about the phospholipids. Sphingophospholipid. Good. So, okay, it takes so long to write these. Okay, hold on. Sphingophospholipid, and then did you say glycosphingolipid? Then there's one more that is a phospholipid. Starts with a G.
Isn't it sphingoglycolipid?
Is it a sphingoglycolipid?
Yeah.
Okay. Backwards. Okay, so sphingoglycolipid, and then
Glycerol.
yes. Glycerophospholipid. What else? Something that starts with an E. Eicosanoids. What are the 3 eicosanoids? Prostaglandins, thromboxanes, glucotrienes. Yes, good. Okay, what else? Steroids, good. So you can't say membrane lipid. That's not what I'm talking about. When I say class of lipid, I'm talking about it has to be one of these. So for instance, when you guys drew on your pre-lab the structure of cholesterol, I saw 4 rings, do you know what I mean, as part of its structure. And so many of you are telling me, oh, it's a triglyceride. But do triglycerides have a 4-ring structure? No, like they do not. You should be able to identify like something that has 4 rings. There's only one type of lipid that has 4 rings in it. What is it?
Steroids.
It's a steroid. Like, this has to make sense. Do you know what I'm saying? Like, you need to make sure that it makes sense. And really, for your quiz, you definitely need to know the classes of lipids, what they're made of. So are they made of glycerol and an alcohol? Are they made of glycerol and 2 fatty acids? Like, what are they made of? And then what is their function? Okay, so be sure that you know that. All right. So anyway, this brings us to page 102, and I will give you stuff like this on the exam, where I'm going to give you some lipids and ask you to identify, like, what class of lipid are they, what components are they, which ones are going to be saponified, which ones would you find in cell membranes, and so forth. So this is good practice for your exam. Okay, so I think we're with you. Yep, Sarah. So A, which structure is a wax? E as in elephant. Good. And then B, which structure is a salt of a fatty acid? And then soap, Dylan? B, that's right. Which structures can be produced during base catalyzed hydrolysis? So Amy, with an F, we're on C. B. Yep, I agree. And then which structure besides A can undergo base catalyzed hydrolysis? So Amy, C and D. Could E undergo hydrolysis? A wax?
Yeah.
It definitely can, right? So you're looking for things that have ester and amide linkages. So for D, C, D, and E, all 3 of those can undergo saponification. Okay, which structure contains glycerol? So this is Amy with an L.
Oh, she went the first time.
Oh, you went the first time. Okay, then Amy with a P.
C
You're like all in a row right there, right?
and
Did you just say C?
C and D.
C and D, I agree. And then which structure would you find in cell membranes? That is for you, Leah? D. D? I agree. Why not C? What kind of lipid is C?
Triglyceride.
Triglyceride, and that is never found in cell membranes. Okay, good. That's good practice. All right. So then flip back to your lab. A couple of things. I gave you back your pre-lab, and if you could, for a moment, flip to page 108, question 5. So of your pre-lab. And it was a question about compare a fat versus an oil. How are they similar and how are they different? So this is a good test question as well. And I'm looking for some specific things here. So if we're talking about fat versus oil, I want you to tell me, are they saturated or are they unsaturated? I want to know, are they from plants or are they from animals? Okay, I want to know, like, who has the higher melting point? Who has the lower melting point? So make sure that you're telling me about melting point. And that's good. But some of you are missing something. So if I circled something in there, maybe you missed something along those lines. Maybe you told me it was saturated versus unsaturated, but didn't say if it was from a plant versus an animal or who had the higher melting point. Those are good questions to ask on your quiz too, or in your test. Okay, and then the rest of it I think you guys can kind of figure out. Let's talk about the lab. Maybe before we get too going, let's look at one more thing, page 113. So look on page 113. So this lab is kind of involved. Page 113 is something that you're going to do at home. So I just want to talk about it for a second, and then that way you're kind of set up and ready to go. What I want you to do is find 3 foods at home or at the store, wherever you go, or just Google it. That's fine too. But I want you to find 3 foods that are related. So like if you picked, Cheetos, then you would find like Cheetos and maybe 2 other kinds of chips or whatever. Maybe you want to do 3 cereals. Maybe you want to do like 3 crackers. Make sure that they're related, and make sure that when you look at the food label that it lists carbs, fat, and protein. Okay? So if it's like, don't pick, say, Starburst, because the only thing that is in that is sugar. Do you know what I mean? Like there's no fat and protein in that. I want you to find something that has has a little of all 3. So you could do granola bars. Like I don't really care what you do, whatever you like. Anyway, and the purpose of this is to figure out, like, off of the food label, you're going to copy down the grams of fat, the grams of protein, and the grams of carbs, okay? And so there's a spot for you to write that down in. So, like, if we were going to do it for Cheetos, I would write down the serving size. We also actually write down how many calories per serving. But I would write that there's 10 grams of fat, and there's 13 grams of carbs, and there's 2 grams of protein. And so maybe you do this with, whatever you want. Maybe like, and some of this is kind of interesting too, like sometimes the cereals that you think are so healthy for you, after you do the calculations, you might think, oh, I didn't realize there was that many, how much of the calories come from, say, fat or from protein or whatever. Some of the cereals you might think that are so terrible for you, like Fruit Loops, may not be as bad as you think. I'm not saying that's true. I just made that up. Do you know what I mean? Fruit Loops may not be that healthy. But anyway, but my point is it's kind of interesting. So you'll get the grams from the food label. Then do you see at the top of the page, it tells you how many calories come from every gram of carbs, fat, and protein. So for carbohydrates, there's 4 kilocalories per gram. Fat's 9 kilocalories, and then protein is 4. So I want to see your calculations for this. This is like a Chem 2A calculation. So if I have 10 grams of fat, then I can say that there's 9 kilocalories per gram of fat. And that'll be 90 kilocalories of fat. And then you'll do the same thing for carbs. For carbs, it's 4 kilocalories per gram. gram. And so that'll be whatever that is, 52. And then you do the same thing for your protein. I think that's still 4. And then that'll be 8. And then you're gonna add them all together. Okay, so I add them together, that'd be like 150. Okay, and that's what I get for my calculation. Then I compare that to what I see on the bag. On the bag, it says 150 calories per serving. So that matches, they should be close. Sometimes when you do these calculations, maybe you calculate 152, and what you see on the food label is 157. But they should be close, do you know what I mean? You're doing something wrong if you calculate your total to be 150 and the food label says 370. Do you know what I mean? Then something went wrong. Do you know what I mean? They should be really close. And then the last part of it asks you to do the percentages. And please make sure you show your work. So you're gonna find the percentage of calories from carbohydrates. That means I'm gonna take the calories from carbohydrates, so the 52 kilocalories. And I'm going to divide it by the total kilocalories that I calculated, and then times 100. Okay, and then that will give me my percent. And then you'll do that for each of the 3 food groups. Okay, so of the fat, the protein, and the carbs. And then after you've done it for each of your 3 foods, then you'll say, hey, this is the one that I think is the healthiest, and then tell me why. Does that make sense? So you can do that at home. It's just a calculation, but I want to make sure that I explain it, make sure we're ready to go on that. Okay, so let's talk about the lab. Be with me on, let's see, page 109. So there's 3 parts to the lab, and probably what you want to start with is part 1, just because it's going to take a little bit of time. So you're going to need a hot plate, and when you do part 1, there's sort of, you're going to test 4 different fats. So we're testing butter, margarine, light margarine, and then Crisco. And all we're really trying to find out is how much fat is in each one of them versus how much water. Which one do you think is gonna, at least of these 3, butter, margarine, and light margarine, which one do you think will have the most fat? Butter. And then probably light margarine will have the least. It really should make sense when you do these calculations. So part 1, you do need to do the calculations for. So the first thing to do is get a hot plate going of water. It's going to take a while for that water to heat up. And then this lab is a little bit messy. I mean, this is probably the grossest part of the lab. So you're going to need a centrifuge tube. I'm going to have you pick just 2. So maybe you and your partner do butter and margarine, and then you get with another group and they do light margarine and Crisco. Does that make sense? So you're only going to do 2 yourself, and the other group is going to do the other 2. Then you guys will collect data and make sure you do the calculations, all right? And they should make sense. So in your data page, on page 109, do you see where it says mass of empty centrifuge tube? Does everyone see that? So that is these. You're using these tubes. They have a pointy tip. They're on the cart. You're not using your own test tubes. Okay, so you weigh this empty, and then you're going to squirt in whatever your fat is. So if you're doing the butter and the margarine or whatever 2 it is you're doing, I think you need like 4 to 6 grams. Oh, 6 to 8. That's fine. So the fat is in these squirt bottles, and they're pretty soft. You probably have to like tap them down a little bit. But my advice is weigh this empty and then squirt in the fat. And a couple of things, like they sometimes will have like an air pocket and all the fat is going to be up here. That's okay. So about 6 to 8 grams. If you get a little less than that, a little more than that, it's fine. Like don't mess around with this. Do you know what I mean? Just get as much in there as you can. You know, maybe half fill it, weigh it, and see about where you're at. And if half full is pretty good, then half full for the rest of them. Do you know what I mean? If it starts to be really clogged and you need a little more, you can try to tap them a bit on the countertop, and that will help them come down. So anyway, about 6 to 8 grams of each one, and then weigh them a second time. So you're weighing this empty, you're squirting the lipid in there, and then you're weighing it again. And that will allow you to figure out how much of the butter or margarine or whatever it is that you have in there. Does that make sense? And then drop them into your water bath. As they heat, everything should melt down. Do you know what I mean? And so the goal is to get them liquid, however long that takes. So while you're waiting for your water bath to heat and for these to kind of dissolve, do you know what I mean, or at least become liquid, then you can set up some of the other parts of the lab. Does that make sense? But get this one started. Once they're done, so they're all melted, then you're going to centrifuge them. And the point of this is to just get the water and the fat that's in the lipid to separate. And so you need these centrifuge tubes to be balanced. So that means, like, if I did butter and margarine, when I hold them next to each other, they should have about the same amount in there. If they don't, let's say one's really full and the next one's not, then get a separate tube and just fill it with water so it's matching. Does that make sense? They need to be fairly close to each other. If they are not, when you go to centrifuge, the centrifuge, well, you can break it. So, like, let's make sure that they're balanced. And then when you put your centrifuge tubes in, they go in opposite of each other. So, like, you sort of will put them in kind of like this. Do you know what I'm saying? And there's 6 slots, so you can spin up to 6 tubes. So you might have your 2 tubes, and there might be another group that's ready to spin too. And so you go ahead and stick them in, turn the lid. There's a little, like, twisty part to lock it, so you can't lift the lid up. And then you turn it on. And it should sound like that. Do you guys hear it? If it sounds not like that, it is not balanced. Do you know what I'm saying? And I've actually had these walk across the countertop because they were so unbalanced. So if that happens, just turn it off and wait. And if it really does start walking, like, you can hold on to it. Do you know what I'm saying? But you guys should balance it. Like, that's the whole point, so it doesn't happen. So you don't have to let them spin too long. I let this spin for maybe 10 seconds, and then I turn it off. And notice, do not open the lid until it's done. When are you going to know if it's done? Well, you don't hear it anymore. Plus, there's like a hole in the center. You can look at it and make sure it's not spinning anymore. I don't want you opening it until it's done spinning, okay? Then you just open it and pull your samples out. Do you know what I'm saying? Like, it's simple. They don't have to spin for very long. When you're done, what you should see is maybe some white solid, some yellow part on, or maybe a a clearish part on the bottom, and then yellow on the top. So yellow is fat, and whatever else is left behind, we're gonna call water. Okay, and in some cases, especially for the butter, there's like milk solids that are part of it, but we're still just gonna call that water, okay? So you're gonna suck off the yellow layer, okay? That is on the top layer. So you guys have like little glass pipettes in your locker. Use that to suck out the fat layer, the top layer. And so if you look at your data page, do you see like halfway down, it says mass of tube and water in sample. Does everyone see that? So what that means is after you spin it, you suck off the fat and just squirt it into the sink. That's fine. Suck off the fat, and what's left is just the water, and then re-weigh it. Okay, and so my advice, because it's going to have liquid in there, you don't want to lay it on the balance. So my advice is have a beaker in the balance, tear it, and then put your centrifuge tube inside of it so it doesn't spill. Does that make sense? So you're weighing it again. That will allow you to figure out how much water is in the test tube. So if I subtract the mass of the test tube, I can get the mass of water. And then if I know the mass of the sample and the mass of water, then the difference will be the fat. Does that make sense? And then you're going to do percentages. So this involves calculations. You need to have these calculations done before I'll sign your data and you can leave today. Okay, so it's important that you do those together. You'll need a calculator, so you can use your phone, or I have some you can borrow. All right, that's part one. Let's talk about part two. So part two is going to be different. So these are in test tubes. Okay, in test tubes. You're going to need 5 test tubes. One is your control. One will have butter. One will have cottonseed oil. One will have peanut. And then the last one's just going to have bromine in it. When you start part two, gloves, Okay, that's really important. You need gloves. And then the second thing is, I want you to do it in the hood. So you can get set up here and then take your test tube rack to the hood and do this part of it. Okay, and this part you'll just do with your partner. And so what you'll have is in one test tube a dropper of methylene chloride. So the methylene chloride is in the clear bottle. What I mean is just, suck up as much as you can into the dropper and squirt that into a test tube. That's one dropper full, okay? So one dropper full of the methylene chloride. And then same thing, in the butter, one dropper of methylene chloride, one dropper for the cottonseed, one dropper for the peanut oil. So all 4 of your first test tubes should have one dropper full of the methylene chloride, okay? And then for the butter, you're gonna have 1 drop of butter in there. So the butter needs to be melted. My advice is, while you're waiting for your water bath to heat up, get part 2 set up. And so once the butter is melted for part 1, just take a drop of it out. Does that make sense? And so make sure when you add this drop of butter that it doesn't drizzle down the side of the test tube. So you need to aim it so it goes right into the methylene chloride. All right, so 1 drop of butter in with the methylene chloride, 1 drop of the cottonseed oil, and 1 drop of the peanut, and that stuff's up here. Okay, so just literally 1 drop. Again, aim it so it goes right into the methylene chloride. Okay, and then the other dropper bottle that's up here is just bromine. So in your last test tube, add a dropper full of bromine to this last test tube. And while you're at it, put in 1 drop of bromine just to the first test tube. So it should turn kind of a light orange color. This is your control, okay? Then go back to the hood. Do not take the bromine with you. You have a test tube of bromine that you'll be using. Does that make sense? So leave this for everyone to get all set up. So you'll have your 5 tubes set up, then you'll go back to the hood. And with your glass dropper, you're gonna start adding bromine from the test tube into, say, your butter. And so the test here is to figure out how saturated they are. So bromine, will it react with the alkanes, the ones that are saturated, or with the alkenes, the ones that are unsaturated? Alkenes. So the more bromine you add, the more unsaturated it is, okay? So which one do you think will take the least amount of bromine? Will it be the butter or will it be the oils? Which one do you think is most saturated, meaning will take the least amount of bromine?
Butter.
The butter, and that should make sense. So this can take a while. What you'll do, and you don't have to add them 1 drop at a time. You can do 5 drops at a time. So maybe you drop in 5 drops, shake your test tube. If it's still clear, you're not there yet. So you're adding bromine until, in theory, they get as dark as your control, or at least until they're no longer clear. Do you know what I mean? So your control may be too dark. So I really just want you to take these 3 test tubes till they're no longer clear. So it will be kind of like a light, faint orangey color. Does that make sense? And I would add them 5 drops at a time. I want you to be careful when you add the bromine. It has a tendency to shoot out of your glass pipette. Do you know what I mean? So I think the best thing to do is try to hold your glass pipette vertical, so you know that you're just dropping it, and if they dribble out, it's fine, just as best you can in terms of keeping track of the bromine. So put a few drops in, shake it. If it's still clear, keep adding until it's this faint orangey color. It could take over 100 drops. I've had sometimes them take 150. So I just want you to know, as you're out there, if it feels like it's not happening, it eventually will. Does that make sense? This is why you go 5, maybe even 10 drops at a time. Does that make sense? So the butter shouldn't take too much, probably less than 50, and then the oils will take more than that. Okay, so that's part 2. Make sure you're wearing gloves and it's in the hood. Okay, that's really important. Part 3, and this is the last part. That group that you kind of split butter and the light margarine like you did too and they did too, you're gonna do the same thing. One of you is gonna do an egg, and the other group is gonna do Cheetos, okay? And what we're gonna try to find out is how much lipid is in the egg. And there's definitely lipid, especially what you're gonna take is the egg yolk. So you'll take half of an egg and you'll just take the egg yolk and you need to take a beaker, weigh it empty, and then you'll add the food to it. So we wanna know the mass of the food. So if you look at the data page for this, it's page 110. So look at page 110. Do you see where it says mass of empty beaker without food? Does everyone see that? So you'll weigh the beaker empty, and then you'll add the food to it, and then you'll weigh it again. If you're doing Cheetos, just take like maybe 2 or 3 Cheetos. Probably 2 is good. They're the really big kind. So a couple of Cheetos is fine. If you're doing the egg, then just half of an egg, and we only want the egg yolk. Okay, so it'll be like half an egg yolk. Does that make sense? So you'll weigh the beaker empty, weigh it with the food in there so we'll know how much food is in there. And then I want you to kind of chop it up. So if it's the egg, it's kind of easy. I'll get out the glass stirring rods. You kind of want to crumble up the egg yolk. If it's the Cheeto, stab at it. Do you know what I mean with the glass stirring rod? We want to sort of get it crumbly. And then what you're going to do is squirt in a lot of acetone. So I want it to be like juicy in there. Do you know what I mean? Like slushy, so that you're swirling it and you can see the food moving. So the lipid is not very soluble in water because it's nonpolar, but it is pretty soluble in acetone. So basically what we're doing is dissolving the lipid that's in the food. And then what you'll do is get a piece of filter paper. I'll get those out. You know the filter paper that we used before where you fold it in half and you fold it in half again, make a little cone? And so I just want you to filter the food. Well, actually, let me think about that. Yeah, I do. I want you to filter the food into a clean, dry, pre-weighed beaker. So do you guys see, like, let's see, it says mass of empty beaker with no lipid, like sort of halfway down that first row. That is the beaker. So you'll weigh it empty. Filter into that beaker. Make sure that beaker has you and your partner's name on it. And so at the end, what you'll have is lipid and acetone. But we want just the lipid. So leave it in the hood for the acetone to evaporate by Thursday. Does that make sense? So that you're not going to finish part 3, but you'll have it ready to go for Thursday. Does that make sense? And so the order in which you do these isn't really important, but you do need to get all 3 parts done. Make sure that parts 1 and 3 you're kind of splitting with another group, okay? And then you'll share your data at the end. Does that make sense? So goggles, you'll need your lab coat, you'll need gloves for part 2, and then go ahead and get started. Do you guys want to?
Okay.
We'll do butter and margarine. You guys do like margarine. We'll do butter and margarine.
So I would get my hot plate going. That's going to be the longest part.
Okay.
Yes, bye.